the process by which a duplicate copy of the genetic material is made before a cell divides. The steps in replication are highly conserved in all organisms. There are a few details that differ between bacteria and eukaryotes, but we focus on bacterial replication in this animation for simplicity. The process of DNA replication is broadly described as having three phases: initiation, elongation, and termination. Let's begin by considering the initiation phase. For copying to take place, the double helical structure of the DNA must first be opened at a site termed the origin of replication to allow access by the machinery that will copy the DNA. This opening is regulated by a series of initiation regulatory proteins not shown here. Once open, a DNA helicase, a ring of 6 subunits, is loaded onto one strand of the DNA at each side of the replication bubble. This establishes 2 replication forks that will move away from each other as replication proceeds. The synthesis of DNA is catalyzed by an enzyme called DNA polymerase. However, DNA polymerases cannot start a new strand from scratch. Instead, they can only elongate an existing polymer of DNA or RNA. Consequently, a primer sequence needs to be made from which DNA polymerase can continue its synthesis. Primers are made by an enzyme called primase. Once access to the single strands of the DNA has been obtained, the primase is loaded onto the DNA. The primase synthesizes a small stretch of RNA by copying the sequence of nucleotides on the template strand. A DNA polymerase will later elongate this small RNA stretch. The next steps allow loading of the DNA polymerases onto the DNA to copy the entire chromosome. The first component that is loaded is the sliding clamp, a ring-shaped structure that binds at the 3' end of the newly made RNA primer. To get onto DNA and encircle it, the clamp is loaded by a clamp loader complex. The 5-protein clamp loader complex binds to the sliding clamp, and binding of ATP allows opening of the sliding clamp ring. The opened ring then encircles the template primer junction at the 3' end of the primer. Interaction with the 3' end of the DNA stimulates ATP hydrolysis, and the clamp loader dissociates from the DNA to be replaced by the replicative DNA polymerase. The 3' end of the primer is positioned in the polymerase active site to allow addition of nucleotides to this end. During elongation, the replication bubble gets bigger as the helicases move apart, establishing 2 replication forks that move in opposite directions. The sliding clamp and polymerase unit at each replication fork moves along the DNA as the helicase unwinds the double-stranded DNA to expose single strands. Let's focus on just the right-hand fork for the moment, as the processes at both forks are identical. Polymerases are loaded onto both the top and the bottom DNA strands at the fork. Because polymerases only synthesize DNA in the 5' to 3' direction, the 2 strands must be copied in opposite directions. The bottom strand in this fork is the leading strand. The polymerase on this strand moves continuously from left to right, synthesizing DNA as it travels. The top strand is the lagging strand, on which just a short stretch of DNA is made by the polymerase elongating in the 5' to 3' direction. After synthesizing this short stretch of DNA, the polymerase then dissociates and a new polymerase binds at the fork to elongate the next RNA primer. So priming and elongation are happening repeatedly on the lagging strand as the replication fork moves along the DNA. Synthesis is discontinuous. The short stretches of DNA that are made on the lagging strand are called Okazaki fragments after their discoverer. During the course of replication, these fragments are stitched together. When the lagging strand polymerase runs up against a previously made Okazaki fragment while in the process of synthesizing DNA, the replicative DNA polymerase III is replaced by DNA polymerase I, which has 5' to 3' exonuclease activity. This exonuclease then degrades the RNA primer in front of it, synthesizing DNA as it goes. Once the RNA primer is removed, the 2 sections of DNA are joined by DNA ligase. Now the lagging strand has been elongated by one more Okazaki fragment. While this view of the replication fork is easiest to understand, in reality the leading and lagging polymerases are coupled and move in concert with the helicase. This coupling requires one DNA strand to be looped around so that the lagging polymerase can synthesize DNA in the 5' to 3' direction while moving with the fork. We explore this coupling in more detail in a separate animation. Much less is known about the details of the termination of DNA replication. Once the entire chromosome is copied, the two replication forks meet each other and are dismantled. The ends of the new DNA strands are then joined, again by DNA ligase. The two daughter DNA molecules now consist of one parent strand of DNA shown in grey and one newly made strand shown in green. Thus, DNA replication is termed semi-conservative.
Transkripsi dinyatakan sebagai sintetis RNA dari dasar DNA. Transkripsi DNA adalah proses di mana informasi genetik ditranskripsi dari DNA ke RNA. Transkripsi DNA atau RNA yang ditranskripsi digunakan untuk memproduksi protein. RNA itu sendiri terbagi atas 3. Ada 3 tipe RNA. Yang pertama mRNA, yang kedua rRNA, dan ketiga tRNA. Proses transkripsi DNA dilakukan 3 langkah utama, yaitu inisiasi, elongasi, dan terminasi. Inisiasi adalah transkripsi DNA terjadi di hadapan enzim RNA polimerase. Ada urutan nukleotida spesifik yang mengatur RNA polimerase di mana untuk memulai dan di mana untuk mengakhiri sintetis itu. RNA polimerase menempel pada DNA pada area spesifik disebut daerah promoter. Yang kedua, elongasi. Protein yang dikenal sebagai faktor transkripsi membuka untai DNA untuk memungkinkan enzim RNA polimerase untuk menuliskan untai tunggal DNA menjadi satu untai RNA polimer. Ini untai RNA polimer dikenal sebagai messenger RNA atau mRNA. Untai mRNA berfungsi sebagai template dan disebut untai antisense, dan untai yang tidak ditranskripsi dikenal sebagai untai sense. Tahap ketiga, terminasi. Enzim RNA polimerase bergerak di sepanjang untai DNA sampai menemukan kodon terminator. Kodon adalah 3 untaian basa nukleotida mRNA yang diartikan sebagai asam amino. Pada titik terminator sequence RNA polimerase melepaskan polimer mRNA dan melepaskan diri dari molekul DNA. Adapun transkripsi dalam prokariota dan eukariota. Yang pertama, transkripsi dalam prokariota pada bakteri juga dikenal sebagai transkripsi prokariotik. Ini adalah proses di mana messenger RNA mentranskrip materi genetik pada prokariota. Langkah pertama, inisiasi. Dalam prokariota, proses transkripsi dimulai dengan pengikatan RNA polimerase ke DNA promotor. RNA polimerase mengikat promotor spesifik dalam DNA prokariotik. DNA ini dibuka dan dikenal sebagai kompleks terbuka. RNA polimerase mentranskripsi DNA dan menghasilkan transkrip yang tidak dapat meninggalkan enzim sampai faktor memisahkan dari enzim inti sendiri. Tahap kedua, elongasi. Promotor berbeda dalam kekuatan dan ini adalah bagaimana mereka mendorong transkripsi urutan DNA mereka yang berdekatan. Enzim RNA polimerase menambahkan nukleotida ke ujung 3 dan membangun rantai polinukleotida, dan ini melibatkan pengembangan hamparan DNA yang beruntai tunggal. Tahap terakhir, terminasi. Pada prokariota, pemutusan dapat dilakukan dengan 2 cara yang berbeda, yakni terminasi intrinsik dan terminasi row dependent. Terminasi intrinsik adalah di mana transkripsi dihentikan ketika RNA yang baru disintetis membentuk 9C seperti rambut lingkaran yang jenis row tergantung dari terminasi. Faktor protein yang kenal sebagai row mendestabilkan interaksi antara template dan kemudian mRNA melepaskan mRNA yang baru disintetis. Kemudian transkripsi pada eukariota dilakukan dengan langkah berikut. Yang pertama, inisiasi. Dalam inisiasi, transkripsi eukariota membutuhkan kehadiran urutan promotor inti dalam DNA. Daerah pada promotor eukariota ditemukan bagian hulu atau upstream dari situs transkripsi. RNA polimerase berkaitan dengan promotor inti untuk memulai transkripsi. Proses transkripsi lebih kompleks pada eukariota. Sekelompok protein yang disebut faktor transkripsi memediasi pengikatan di RNA polimerase dan memulai transkripsi. Yang kedua, elongasi. RNA polimerase melintasi untai cetakan dan menambah basa untuk melengkapi template DNA dan membuat salinan RNA. Elongasi pada eukariota juga melibatkan mekanisme proofreading yang menggantikan basa yang salah ditempatkan. Yang terakhir, terminasi. Pemutusan proses transkripsi pada eukariota kurang dipahami. Ini melibatkan pemecahan transkrip RNA yang baru disintetis yang diikuti oleh penambahan seperti yang independen dari template dan ujung baru 3.
Arrangement of amino acids in a linear specific sequence and formation of polypeptide chain according to the specific sequence of information written on mRNA is called translation, changing the code language on mRNA into the language of amino acids. Translation occurs in the cytoplasm. The primary requirements for translation process are ribosomes, mRNA, tRNA, amino acids, enzymes, ATP, GTP, and specific activators. The process of translation involves 1. Activation of amino acids, its transfer to tRNA. 2. Initiation of polypeptide chain. 3. Elongation of polypeptide chain. And 4. Chain termination. 1. Activation of amino acids and its transfer to tRNA. Amino acids are activated by ATP in the presence of an enzyme known as amino-acyl synthetase and linked to the cognate tRNA, a process called as charging of tRNA or amino-acylation of tRNA. As a result, amino-acyl tRNA is formed, which moves towards ribosomes. AA, amino acid, and ATP, an enzyme, amino-acyl synthetase, gives AA and AMP, an enzyme complex, and PP, pyrophosphate. AA, AMP, enzyme complex, and tRNA gives AA, tRNA complex, AMP, and enzyme, amino-acyl tRNA. 2. Initiation of polypeptide chain. AUG is initiation codon of mRNA. It codes for methionine. Formyl group is attached to free amino group of methionine. The tRNA bringing formyl methionine is called fmet tRNA, and it acts as initiator. As the anticodons of tRNA are complementary to the codons of mRNA, they can develop loose hydrogen bonds with codons of mRNA. Ribosomes have two sites for tRNA. 1. Amino-acyl or acceptor site, or A site. 2. Peptidyl site, or P site in the larger subunit. Amino-acyl tRNA first binds to A site of ribosomes and move to P site, while initiator tRNA first binds to P site. The initiator amino-acyl tRNA in the presence of mRNA combines with subunits of ribosomes. This process requires energy. Elongation of polypeptide chain. Chain elongation takes place by the establishment of peptide bonds between the amino acids as the ribosomes move along mRNA one codon at a time in 5' to 3' direction. Depending on the second codon of mRNA, the tRNA with anticodon complementary to the codon of mRNA, forms a complex with the corresponding activated amino acid and moves to the ribosomes and binds at the A site. This is facilitated by elongation factor and energy in the form of GTP. 4-methionine in P site establishes a peptide bond with the second amino acid at A site. Polypeptide chain grows in C to N direction. Carboxyl group of first amino acid and amino group of second amino acid. Peptide bond is catalyzed by the enzyme peptidyl transferase. After the bond formation, ribosomes move to the next codon of mRNA. This movement of ribosomes relative to mRNA, resulting in the shift of peptidyl tRNA from A site to P site, is called translocation. Translocation requires translocase and GTP. Therefore, aminoacyl tRNA enters A site and moves to P site after developing peptide bond. 4. Chain termination. When the terminating codons UAA, UAG, or UGA present in mRNA, it indicates the end of polypeptide synthesis. They do not code for any amino acid, and hence no amino acid enters A site, and thus they act as stop signals. At the end, a relative factor binds to the stop codon, terminating translation and releasing the complete polypeptide from the ribosome.